Let's begin with phrenology, a once popular theory that claimed bumps on the skull could reveal our mental abilities and character traits. Although it captured public imagination in the 19th century, phrenology was ultimately debunked. Its legacy, however, sparked curiosity about the brain's structure and function, paving the way for modern neuroscience. Today we know that personality and intelligence are not determined by skull shape, but by complex neural networks within the brain. This transition from pseudoscience to scientific inquiry set the stage for deeper exploration into how our brains truly work. Neurons are the fundamental information processing cells of the nervous system. The human brain contains roughly 86 billion neurons, each forming thousands of connections. These cells are responsible for receiving, integrating, and transmitting signals throughout the body. Their intricate web of connections forms the basis for all our thoughts, feelings, and actions. Understanding the structure and function of neurons is essential to grasp how the brain operates. As we move forward, we'll break down the key components that make each neuron a powerhouse of communication. Cell body, or soma, is the metabolic center of the neuron. It contains the nucleus and most organelles, integrating incoming signals from other neurons. If the soma is destroyed, the neuron dies, highlighting its critical role in maintaining cell health. The soma's ability to process and combine signals is essential for the neuron's function. This integration determines whether the neuron will pass along information to other cells. As we explore the neuron's structure, the soma stands as the hub where life and communication converge. Dendrites are highly branched extensions that receive signals from other neurons or sensory receptors. Their extensive branching increases surface area, allowing for more input. Dendritic spines, small protrusions on dendrites, form synaptic contacts and play a crucial role in learning and memory. By collecting incoming information, dendrites ensure that neurons stay connected and responsive to their environment. This network of branches is vital for the brain's adaptability and communication. Axon is a single long fiber that conducts electrical impulses away from the soma toward other neurons, muscles, or glands. Axons can range from less than a millimeter to over a meter in length, especially in motor neurons extending from the spinal cord to the toes. This structure is essential for transmitting information rapidly and efficiently throughout the body. The axon's ability to carry action potentials makes it the main highway for neural communication. Axon hillock is the junction between the soma and the axon. It acts as the trigger zone where action potentials are initiated when the summed input exceeds the threshold. This region is crucial for determining whether a neuron will fire, making it a key player in neural decision-making. The axon hillock's role in integrating signals and triggering action potentials ensures that information is passed along only when necessary. Myelin sheath is a fatty insulating layer formed by oligodendrocytes in the CNS or Schwann cells in the PNS. Myelin dramatically increases the speed of signal conduction by enabling saltatory conduction. This insulation allows electrical impulses to jump from node to node, making communication faster and more efficient. Diseases that damage myelin, such as multiple sclerosis, can slow or block signal transmission, leading to various neurological symptoms. Nodes of Ranvier are small, unmyelinated gaps between segments of myelin where ion channels are concentrated. The action potential jumps from node to node, allowing for rapid and energy-efficient signal transmission. These nodes are essential for maintaining the speed and direction of neural communication. Their unique structure ensures that the neuron can quickly restore its resting potential after each impulse. Axon terminals, also known as terminal buttons or synaptic boutons, are bulb-shaped endings of the axon that contain synaptic vesicles filled with neurotransmitters. When an action potential arrives, these vesicles release their contents into the synaptic cleft, allowing communication with other neurons or target cells. This process is fundamental to how the nervous system transmits signals and coordinates responses. Let's recap the key structures and their functions. Dendrites receive incoming signals, the soma integrates them, the axon hillock initiates action potentials, the axon conducts signals away from the soma, the myelin sheath insulates and speeds conduction, nodes of Ranvier are sites of ion exchange, and axon terminals release neurotransmitters. Each component plays a unique role in the neuron's ability to communicate and process information. Neurons are classified by the direction they carry signals. Sensory afferent neurons bring information from sensory receptors to the CNS. Motor efferent neurons carry commands from the CNS to muscles and glands. Interneurons, found entirely within the CNS, connect sensory and motor neurons and are critical for complex functions like thinking and learning. Each type has unique structural features and plays a vital role in the nervous system. Sensory neurons carry information from sensory receptors to the central nervous system. Most are unipolar, meaning they have a single process that splits into two branches. For example, a sensory neuron in your fingertip detects heat and sends the signal to your spinal cord and brain. These neurons are essential for bringing external information into the nervous system, enabling us to perceive our environment. Motor neurons carry commands from the CNS to muscles and glands. These neurons are multipolar with many dendrites and one axon. For instance, a motor neuron sends a signal from the spinal cord to a leg muscle, causing contraction. Motor neurons are responsible for executing voluntary movements and responding to the brain's instructions. Interneurons are found entirely within the CNS and make up about 99% of all neurons in the body. They connect sensory and motor neurons, processing and integrating information. Interneurons are multipolar and are critical for complex functions like thinking, learning, and decision-making. Their abundance and versatility make them central to the brain's ability to handle sophisticated tasks. When a neuron is not firing, it maintains a stable electrical charge called the resting potential, approximately -70 millivolts. The inside of the neuron is negative relative to the outside. This state is maintained by the sodium-potassium pump, potassium leak channels, and large negatively charged proteins trapped inside the cell. The resting potential represents stored potential energy, ready to be released when the neuron fires. Action potential is a rapid, temporary reversal of the electrical charge across the neuron's membrane. It is triggered when a stimulus causes depolarization to reach the threshold. The action potential travels down the axon, allowing the neuron to communicate with other cells. This electrical signal is fundamental to how the nervous system operates, enabling quick and precise responses. Action potential unfolds in several stages: stimulus and threshold, depolarization, repolarization, hyperpolarization, and return to resting potential. Each stage involves the opening and closing of specific ion channels, allowing sodium and potassium ions to move in and out of the cell. This sequence ensures that the neuron fires efficiently and resets for the next impulse. All or none principle states that a neuron either fires a complete action potential or does not fire at all. There is no partial action potential. Once the threshold is reached, the action potential always has the same magnitude regardless of stimulus strength. The nervous system codes stimulus intensity by the frequency of firing and the number of neurons activated. Immediately after an action potential, the neuron enters an absolute refractory period, during which no stimulus can trigger another action potential. This ensures the signal travels in one direction. The relative refractory period follows, where a stronger than normal stimulus can trigger another action potential. These periods set an upper limit on firing rate and maintain orderly communication. Antagonists block a neurotransmitter's functioning. For example, botulinum, a poison found in improperly canned food, causes paralysis by blocking acetylcholine release, which is crucial for muscle contraction. If its transmission is blocked, muscles cannot contract, leading to paralysis. Understanding antagonists helps explain how drugs and toxins affect the nervous system. The brain produces its own naturally occurring opiates called endorphins. These neurotransmitters are released in response to pain and vigorous exercise, helping explain good feelings like the runner's high and the pain-killing effects of acupuncture. Endorphins play a vital role in mood regulation and pain management, showing how the brain adapts to stress and injury. Synapse is the junction between two neurons or between a neuron and a target cell. The tiny gap between them, called the synaptic cleft, is where neurotransmitters are released. Synaptic transmission involves the arrival of an action potential, opening of calcium channels, release of neurotransmitters, and binding to receptors on the postsynaptic membrane. This process is essential for neural communication. After neurotransmitters have produced their effect, their action must be terminated. This occurs through reuptake, enzymatic degradation, or diffusion. Reuptake involves the presynaptic neuron reabsorbing the neurotransmitter. Enzymatic degradation breaks down the neurotransmitter, and diffusion allows molecules to drift away. Many psychiatric medications target these mechanisms, affecting mood and behavior. Myelinated axons, the action potential jumps from one node of Ranvier to the next, a process called saltatory conduction. This method is much faster and more energy efficient than continuous conduction. Diseases like multiple sclerosis, which damage myelin, can slow or block signal transmission, leading to muscle weakness and cognitive impairment. Neurotransmitters can produce excitatory or inhibitory effects. Excitatory postsynaptic potentials, EPSPs, depolarize the membrane, making it easier for the neuron to fire. Inhibitory postsynaptic potentials, IPSPs, hyperpolarize the membrane, making it harder for the neuron to fire. The balance of these signals determines whether an action potential will occur. Neural integration involves temporal and spatial summation. Temporal summation occurs when multiple signals from the same neuron arrive in rapid succession. Spatial summation happens when signals from multiple neurons arrive simultaneously. The axon hillock sums all EPSPs and IPSPs, and if the net result reaches threshold, an action potential fires. The somatic nervous system enables voluntary control of our skeletal muscles. Also called the skeletal nervous system, it allows us to move and interact with our environment. This system is responsible for conscious actions such as walking, talking, and writing, connecting the brain's commands to our muscles. Autonomic nervous system controls glands and muscles of internal organs, influencing functions like heartbeat, digestion, and glandular activity. It operates automatically, regulating vital processes without conscious effort. This system is divided into sympathetic and parasympathetic branches, each with distinct effects on the body. Sympathetic nervous system arouses and expends energy. When you are alarmed, enraged, or challenged, it accelerates your heartbeat, raises blood pressure, slows digestion, raises blood sugar, and cools you with perspiration. This system prepares you for action, making you alert and ready to respond to stress. When stress subsides, the parasympathetic nervous system produces opposite effects. It conserves energy by calming you, decreasing your heartbeat, lowering blood sugar, and promoting relaxation. This system helps restore balance and maintain homeostasis after periods of excitement or stress. Central nervous system, CNS, consists of the brain and spinal cord. It serves as the body's command center, receiving sensory input from the peripheral nervous system, interpreting information, and coordinating motor output. The CNS is encased in bone for protection, reflecting its critical importance. CNS processes and integrates information, while the peripheral nervous system, PNS, carries information to and from the CNS. The CNS is protected by the skull and vertebral column, emphasizing its vital role. The PNS includes cranial nerves, spinal nerves, and ganglia, connecting the CNS to the rest of the body. Damage to specific brain areas can disrupt vision, speech, or other functions. For example, injury to the back of the brain affects vision, while damage to the left front part impairs speech. These observations helped scientists map the brain's functional regions and understand how different areas contribute to behavior. Electrical activity in the brain sweeps in regular waves across its surface. An electroencephalogram (EEG) amplifies these waves, while a PET scan shows brain activity by tracking glucose consumption. Active neurons are glucose hogs, and PET scans reveal which areas are most active during various tasks. These tools help scientists study brain function in real time. MRI uses a strong magnetic field to align spinning atoms in brain molecules, providing detailed images of brain tissue. Functional MRI (fMRI) reveals both structure and activity by tracking blood flow to active regions. These imaging techniques allow scientists to study the brain's anatomy and functioning without invasive procedures. During embryonic development, the brain forms from three primary vesicles: forebrain, midbrain, and hindbrain. The forebrain includes the cerebral cortex, basal ganglia, hippocampus, and amygdala, responsible for higher-order functions. The midbrain handles visual and auditory reflexes, while the hindbrain manages motor coordination and vital autonomic functions. Brainstem connects the cerebrum and cerebellum to the spinal cord. It includes the midbrain, pons, and medulla oblongata. The reticular formation, a network of neurons, filters incoming stimuli and relays important information. The brainstem controls heartbeat, breathing, and sleep-wake cycles, making it essential for survival. The thalamus acts as the brain's sensory switchboard, receiving information from all senses except smell and routing it to higher brain regions. This egg-shaped structure sits atop the brainstem and plays a crucial role in processing sensory input and directing it to appropriate areas for interpretation. Cerebellum, meaning little brain, enables nonverbal learning and memory. It helps us judge time, modulate emotions, and discriminate sounds and textures. The cerebellum also coordinates voluntary movement, ensuring smooth and balanced actions. Its role is vital for everyday tasks and motor skills. Limbic system sits at the border between the brain's older parts and the cerebral hemispheres. It includes the amygdala, which influences aggression and fear, and the hypothalamus, which regulates hunger, thirst, body temperature, and sexual behavior. The limbic system is central to emotion, motivation, and bodily maintenance. Spinal cord extends from the base of the skull to the lumbar region, about 43–45 cm in adults. It contains gray matter cell bodies, dendrites, interneurons, and white matter myelinated axon tracts. The spinal cord receives sensory information, processes it, and coordinates motor output, serving as a vital communication link. Spinal reflex is an automatic, rapid response to a stimulus processed at the spinal cord level. The reflex arc includes 5 components: receptor, sensory neuron, integration center, motor neuron, and effector. Examples include the knee-jerk reflex and withdrawal reflex, which protect the body from harm. CNS is protected by 3 systems: meninges, 3 membrane layers, cerebrospinal fluid (CSF), and the blood-brain barrier (BBB). Meninges cushion the brain, CSF removes waste and transports nutrients, and the BBB selectively permits passage of substances, blocking pathogens and many drugs. Neuroplasticity is the brain's ability to reorganize its structure and function in response to experience, learning, or injury. Structural plasticity involves growth of new dendritic spines and synapses. Functional plasticity allows reassignment of functions, and synaptic plasticity changes synaptic strength. Plasticity is greatest in early development but continues throughout life. Glial cells outnumber neurons and perform essential support functions. Astrocytes maintain the blood-brain barrier and regulate ion concentrations. Oligodendrocytes form myelin in the CNS, microglia act as immune cells, and ependymal cells line ventricles. In the PNS, Schwann cells form myelin and satellite cells regulate the microenvironment. Glial cells are crucial for brain health and function.